This video is part of a lecture called Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Steps in plant breeding. All plant breeding programs and protocols follow the same few steps with slight variations depending on the species, the traits of interest, and breeding methods that are used. The first step is to collect or otherwise dispose of sufficient genetic variation to work with. This can be as simple as having a genetically diverse population in the field. These days, however, plant breeders dispose not only of the variation in the field, but also have access to seed banks and germplasm collections in which thousands of plant genotypes are stored from all around the world. With the development of new plant breeding techniques and genetic engineering, the available genetic diversity has breached the barriers of the species, and nowadays plant breeders can also search global gene banks for genes of interest of any species that can be introduced in the plant. Once a source of genetic variation is available, a plant breeder needs to identify one or several traits of interest that they would like to improve. This might seem like a straightforward process, but in reality it requires a great knowledge of the plant's genetics, physiology, ecology, and molecular biology in order to guarantee the success of the breeding program. For example, if a breeder is trying to improve a trait which is highly influenced by the environment, the best approach might not be to modify the trait through plant breeding, but through an improvement of the environmental conditions in which the crop is grown. See the heritability video for more details on this. Next, the trait of interest need to be inserted in the crop species that are targeted by the breeding program. Insertion might not be the most appropriate word for all breeding techniques, because this step covers a vast array of approaches that range from simple mass selection, where nothing is inserted in the plant, and instead plants with traits of interest are preferentially reproduced, to actual insertion of genes of interest via genetic engineering techniques, to genome editing, where the plant genome is modified without any external sources. After the insertion of the trait of interest, many breeding techniques would also require to remove any undesired traits that were introduced at the same time. This can be either whole portions of the genome, in the case of conventional crosses, or removing reporter genes that are used to trace the successful transformation of plants via genetic engineering. Trait of interest is inserted, the modified crop needs to undergo a field testing. The testing will confirm that the desired trait is indeed incorporated in the genome and is consistently expressed in the new crop variety, and the safety of the crop for the consumer and the environment. The cycle of identifying new traits to field testing can be repeated several times if needed. In particular, in conventional breeding techniques, 6 to 10 cycles are usually required to achieve new crop varieties that only possess the traits of interest and no undesired traits. Note that the exact steps of the breeding process can vary depending on the type of crop, the traits of interest, and the techniques that are used. In mass selection, for instance, identifying and inserting traits of interest is done during the field testing, when all available and genetically diverse crops are grown in the field, but only the crops with desired traits are left to reproduce for the next year. On the other hand, genome editing does not require removal of undesired traits because the plant genome is modified directly without inserting any foreign genes in it.